Welcome back to the deep dive. Today, we're going to take a journey, into the fascinating world of FTIR spectroscopy.
Oh, very cool.
It's kind of like having a molecular fingerprint scanner, right?
Oh.
It helps us unlock the secrets of protein structure. FTIR stands for Fourier-Transform Infrared Spectroscopy.
Right.
And don't worry, if you're not familiar with it, we're going to break it down, step by step. So think about it this way. Each musical note has a specific frequency.
Yeah.
Well, molecules, they vibrate at specific frequencies too. And FTIR, it uses infrared light to kind of analyze those vibrations.
That's really remarkable. When infrared light hits a molecule, some of it gets absorbed at specific frequencies depending on the molecule's structure.
Okay.
It's like each molecule has its own, unique set of frequencies where it resonates.
So it's kind of like how a wine glass will shatter if you hit the right note.
Yeah, exactly.
But here we're talking about molecules absorbing those specific wavelengths of light.
Yeah, and by measuring which frequencies are absorbed, we get a unique fingerprint for each molecule.
Right.
And this fingerprint, it tells us what the molecule is, but it also reveals clues about how its atoms are arranged.
It's structure.
Its structure, exactly.
Okay, so how does this molecular fingerprint scanner actually work?
Okay.
I mean, walk us through the process of shining light on something and getting a spectrum.
Sure. So the instrument shines a beam of infrared light through the sample and measures how much light passes through at each frequency. This raw data is called an interferogram.
Okay.
Now, an interferogram is a pretty messy signal.
Right.
But that's where the magic of Fourier transformation comes in.
Okay.
It's a mathematical tool that converts the interferogram into a spectrum that's clear and interpretable.
So Fourier transformation takes that jumbled signal and makes it into something that we can understand.
Exactly.
Kind of like taking a blurry photo and bringing it into focus.
That's a good analogy, yeah.
Okay.
So the resulting spectrum shows us those characteristic peaks where light was absorbed. And those peaks, they hold the key to understanding the molecule's structure.
Now, before we dive deeper into, protein structure analysis, I'm curious, what are some of the real-world applications of FTIR?
Oh, FTIR is incredibly versatile.
Okay.
It's used in a wide range of fields, from forensics to environmental science to pharmaceuticals.
Wow.
It's used to identify unknown substances, determine the purity of samples, and even analyze ancient artifacts.
It's amazing how this one technique can be applied in so many different ways.
Yeah.
But for this deep dive, we're going to focus on how FTIR helps us understand proteins.
Yeah.
So we've got our spectrum.
Yes.
With all these peaks and valleys.
Right.
How do we make sense of this data?
Well, the first step is data analysis.
Okay.
We need to make sure we're looking at a clean signal.
Right.
So we start with something called baseline correction to eliminate any unwanted background noise.
Okay.
This makes it easier to see the real peaks that we're interested in.
So it's like cleaning a window to get a clearer view. We're removing the smudges to see the information more clearly.
And then we often use a technique called peak deconvolution,
Okay.
especially when peaks are overlapping and difficult to analyze individually.
I remember seeing a video about deconvolution on the Safe Physics YouTube channel.
Yeah?
They talked about how it's like separating ingredients in a smoothie.
Right.
You can isolate each component to get a clearer picture of what's inside.
That's a great analogy.
Yeah.
Deconvolution helps us tease apart those overlapping peaks so we can examine them individually and extract more information.
Okay, that makes sense. But why is deconvolution so important when it comes to understanding protein structure?
Well, proteins are complex molecules with many different types of bonds and functional groups, and this can lead to a lot of overlapping peaks in the FTIR spectrum, especially in the region called the amide I band.
Okay.
This band is particularly sensitive to the protein's secondary structure.
Now, I remember learning about protein structure in biology class, but I need a refresher. What exactly is secondary structure?
Secondary structure refers to the local folding patterns of the protein chain.
Okay.
The two most common patterns are alpha helices and beta sheets.
So the secondary structure is like the basic building blocks of a protein's overall 3D shape.
Yes.
And you're saying the amide I band tells us about these structures.
That's right.
No.
The exact position and shape of the amide I band is influenced by those hydrogen bonds that hold alpha helices and beta sheets together. So by analyzing the amide I band, we can get clues about which secondary structures are present in the protein.
So it's like those hydrogen bonds are whispering secrets to us through the FTIR spectrum.
That's a great way to put it.
So by carefully analyzing the amide I band, maybe even using deconvolution to see the fine details, we can start to understand how the protein is folded.
Exactly. And understanding the secondary structure is a crucial step in deciphering the protein's overall three-dimensional shape and ultimately its function.
This is all really cool, but I have another question. Can FTIR detect changes in the protein structure?
Absolutely.
Like what if the protein's environment changes or it binds to something?
Proteins are dynamic molecules constantly interacting with their surroundings, and
Yeah.
an FTIR can capture these dynamic changes. For
Right.
instance, changes in temperature, pH, or binding to other molecules can all induce shifts in the protein structure.
Yep.
And these shifts will be reflected in the FTIR spectrum, especially in the amide band.
So we're not just getting a static snapshot of the protein.
Yeah.
We're actually seeing it move and change in response to its environment.
That's right. And one of the most powerful techniques for studying these dynamic changes is called hydrogen deuterium exchange, or HD exchange.
Okay, I'm intrigued. Tell me more about this HD exchange and how it helps us study protein dynamics.
Well, HD exchange involves replacing some of the hydrogen atoms in the protein with deuterium,
Right.
a heavier isotope of hydrogen. The rate at which this exchange happens is influenced by the protein's structure and its environment.
Okay.
For example, hydrogen atoms that are buried deep within the protein core or involved in strong hydrogen bonds will exchange much more slowly than those on the surface or in more flexible regions.
So it's like we're tagging the protein with these deuterium atoms and watching how quickly they trade places with the regular hydrogens.
Exactly.
And the rate of exchange tells us something about how accessible different parts of the protein are.
That's right. And by combining HD exchange with FTIR, we can actually track these changes over time.
Okay.
Remember how I said that deuterium and hydrogen absorb infrared light slightly differently?
Yeah.
Well, we can use that difference to monitor how much deuterium has been incorporated into the protein and where.
This is all blowing my mind. It's like we're watching the protein dance.
Yeah.
And those deuterium tags are showing us its moves.
It's a powerful technique for understanding how proteins behave in real time.
Okay, I'm ready for more. Give me some real-world examples of how HD exchange and FTIR have been used together to study protein dynamics.
Well, one fascinating study looked at how a protein called CATMP receptor protein, or CRP, changes its structure when it binds to a specific molecule.
Okay.
By using HD exchange and FTIR, researchers were able to identify specific regions of the protein that became more or less accessible upon binding,
Got it.
providing clues about how this interaction happens at a molecular level.
So they were able to see how the protein adjusted its dance moves to accommodate its partner.
Yes.
That's so cool.
And these kinds of insights are incredibly valuable for understanding how proteins function in biological systems.
This is also fascinating. We've covered a lot of ground here, and I'm really starting to appreciate the power of FTIR
Yeah.
for understanding protein structure and dynamics. But before we go any further, let's take a moment to recap what we've learned so far.
All right, so we started by exploring how FTIR works, how it uses infrared light to generate a unique fingerprint for each molecule. We talked about the importance of data analysis, especially baseline correction and peak deconvolution in extracting meaningful information from the spectrum. Then we focused on how FTIR, specifically the amide I-band, helps us understand protein secondary structure,
Right.
the alpha helices and beta sheets that are the building blocks of a protein's shape.
Yes.
Finally, we discuss how HD exchange combined with FTIR allows us to study protein dynamics, how proteins move and change in response to their environment and interactions with other molecules.
That was a great summary. And, it's amazing how all this incredible science started with a simple observation, that molecules absorb infrared light in unique ways.
Right.
It's a testament to human curiosity and ingenuity.
Yeah, it really is.
Okay, we've covered a lot in this first part of our deep dive into FTIR, but there's still much more to explore. When we come back, we'll delve deeper into the world of protein structure analysis and discuss some of the cutting-edge applications of this incredible technique. Stay tuned.
Sounds good. It's fascinating how even the slightest change in a protein structure can have a ripple effect on its function. It's a delicate balance.
Absolutely. And speaking of delicate balances, we were just discussing HD exchange,
Right?
and how it can reveal those subtle shifts in a protein structure.
Right. HD exchange combined with FTIR, it's like having a molecular magnifying glass. That allows us to zoom in on those dynamic changes.
So remind me again, how does HD exchange actually work?
Sure.
We're basically swapping out some of the protein's hydrogen atoms with deuterium.
Right, exactly. Deuterium is a heavier isotope of hydrogen.
Right.
And the key is that this exchange process isn't random.
Oh.
The rate at which a hydrogen atom gets replaced by deuterium depends on its environment within the protein.
So hydrogen atoms that are kind of tucked away in the protein's core
Yeah.
were involved in strong hydrogen bonds
Yeah.
are less likely to get swapped out.
Right.
It's like they're shielded from the deuterium in the surrounding solvent.
Precisely. And that's what makes HD exchange so valuable for studying protein structure.
Okay.
It gives us a way to map out which parts of the protein are more exposed or flexible.
Okay, so we've got these deuterium atoms acting like tiny beacons, revealing the protein's hidden movements.
Yes.
But how do we actually detect those beacons? How does FTIR help us track the HD exchange process?
Well, remember how we said that deuterium and hydrogen absorb infrared light slightly differently?
Yeah.
We can use FTIR to monitor those subtle changes in the absorption spectrum.
Okay.
As more deuterium gets incorporated into the protein, we'll see shifts in the spectrum, especially in the amide I band.
So it's like those deuterium atoms are changing the protein's tune ever so slightly,
Yeah.
and FTIR is our finely tuned instrument for picking up those subtle variations.
That's a great way to put it. And by carefully analyzing those spectral shifts, we can determine the rate of HD exchange at different locations within the protein.
This makes me think about how proteins can change their shape when they bind to other molecules,
Right.
like a key fitting into a lock.
Yeah.
Can HD exchange also help us understand those kinds of interactions?
Absolutely. In fact, that study we mentioned earlier, the one with the CRP protein,
Right.
used HD exchange to see how the protein structure changed when it bound to a specific molecule.
Okay.
They found that certain regions of the protein became more exposed while others became less accessible after binding.
So it's like we're watching the protein adjust its dance steps
Yes.
to accommodate its partner.
And these kinds of insights are incredibly valuable for understanding how proteins function in biological systems.
Okay, I have to ask, what are some of the most surprising or unexpected things researchers have discovered using HD exchange and FTIR to study proteins?
Well, one thing that always amazes me is how sensitive these techniques are.
Mm.
We can detect even the tiniest of structural changes, like a single amino acid flipping its orientation within the protein.
So it's like we have a microscopic camera that can capture those subtle movements in slow motion.
Exactly. And those small changes can have a big impact on the protein's function.
I'm also really curious about the role of water in protein structure.
Yeah.
I know water is essential for life, and it must play a big part in how proteins fold and interact. Can HD exchange tell us anything about the interplay between water and proteins?
It certainly can. Remember, the rate of HD exchange depends on how accessible the protein's hydrogen atoms are to the surrounding solvent,
Right.
which is often water.
So if a region of the protein is highly exposed to water, those hydrogens will exchange quickly with deuterium.
Yes.
But if a region is buried or shielded from water, the exchange will be much slower.
That's right. And by analyzing these exchange rates, we can get a sense of how water molecules are interacting with different parts of the protein.
This is all so mind-boggling. It's like we're peeling back the layers of complexity in the protein world, revealing those intricate details that make life possible.
And it's all thanks to these incredible tools like FTIR and HD exchange that allow us to explore the molecular world with such precision.
Okay, we've been focusing a lot on the dynamics of proteins. But I want to shift gears for a moment and talk about their static structure.
Okay.
You mentioned earlier that the amide I band in the FTIR spectrum is particularly sensitive to the protein's secondary structure, those alpha helices and beta sheets.
Right. The position and shape of the amide I band can tell us a lot about the relative amounts of alpha helices and beta sheets in a protein.
So, for example, if we see a strong amide I peak at a certain frequency, we might conclude that the protein has a high proportion of alpha helices.
Exactly. And conversely, if we see a strong peak at a slightly different frequency, it might indicate a dominance of beta sheets.
That's amazing.
Yeah.
It's like we're looking at a musical score and being able to tell what instruments are playing just by analyzing the notes.
It's a beautiful analogy, and just like a skilled musician can hear the subtle variations in a melody, a trained spectroscopist can discern those subtle shifts in the amide I band to gain insights into the protein's structure.
This makes me think about those two proteins mentioned in the Thermo Fisher scientific application note, cytochrome C and concanavalin A.
Yeah.
They have very different secondary structures, right?
Yes. Cytochrome C is primarily alpha helical, while concanavalin A is dominated by beta sheets.
Okay.
And their FTIR spectra clearly reflect those differences.
So even though both proteins are made up of amino acids linked together in a chain, the way those chains fold into alpha helices or beta sheets creates distinct patterns in the FTIR spectrum.
Precisely, and those patterns are what allow us to distinguish between different types of proteins and gain insights into their structure-function relationships.
This is all incredibly insightful. It's amazing how we can use these techniques to study something as tiny and complex as a protein.
It's a testament to the power of scientific inquiry and the ingenuity of the human mind.
Okay, we've explored how FTIR can reveal both the dynamic and static aspects of protein structure.
Right.
But I want to zoom out a bit and talk about the bigger picture. How has FTIR spectroscopy revolutionized our understanding of proteins and their role in biology?
FTIR has been a game changer in protein science. It has provided us with a window into the molecular world that simply wasn't available before.
So we're talking about a paradigm shift in how we study these essential molecules.
Absolutely. Before FTIR, our understanding of protein structure was largely based on techniques like X-ray crystallography, which require crystallizing the protein, a process that can be difficult and time-consuming.
And X-ray crystallography only gives us a static snapshot of the protein, whereas FTIR allows us to study proteins in solution under more physiologically relevant conditions and even observe their dynamics in real time.
Exactly. FTIR has allowed us to move beyond static images and delve into the dynamic nature of proteins, understanding how they fold, unfold, interact with their surroundings, and carry out their vital functions.
And this deeper understanding has undoubtedly led to significant advancements in various fields, right?
Of course.
I'm thinking about medicine, drug development, and even materials science.
You're absolutely right. The insights gained from FTIR have been instrumental in developing new drugs, understanding the mechanisms of diseases, and even designing novel biomaterials.
So we're talking about a tool that's not only expanding our knowledge, but also shaping our world.
Precisely. FTIR is a prime example of how scientific advancements can have a profound impact on our lives.
Okay, before we wrap up our deep dive into FTIR, I want to touch on a topic that's been on my mind, the future of this technology.
Sure.
What are some of the emerging trends or exciting developments in FTIR spectroscopy that we should be keeping an eye on?
FTIR is a rapidly evolving field, and there are several exciting advancements on the horizon.
Okay.
One area of active research is the development of even more sensitive and powerful FTIR instruments, allowing us to study proteins at even lower concentrations and with greater precision.
So it's like we're getting an even sharper molecular fingerprint scanner.
Exactly. And with those sharper scans come even more detailed insights into protein structure and function.
I'm also curious about the role of artificial intelligence and machine learning in FTIR data analysis.
Right.
I imagine these technologies could help us sift through those mountains of data and extract even more meaningful information.
You're spot on. AI and machine learning are already making their mark on FTIR.
Wow.
Helping us automate data analysis, identify patterns, and even predict protein structures. These advancements are paving the way for faster and more efficient protein research.
This is all so exciting. It's clear that FTIR spectroscopy has a bright future ahead, and I can't wait to see what new discoveries it unveils.
The possibilities are truly endless, and who knows what other mysteries of the molecular world FTIR will help us unlock in the years to come.
Well, we've covered an incredible amount of ground today, from the basic principles of FTIR to its groundbreaking applications in protein science. I'm feeling truly inspired by this deep dive, and I'm sure our listeners are too. It's incredible to think that we can now study something, as tiny and complex as a protein with such precision and detail.
It really is remarkable.
We've explored the basic principles of FTIR, how it generates a unique fingerprint for each molecule, and how we can decipher that fingerprint to understand a protein's structure, both its static shape and its dynamic movements.
Right, and we've seen how techniques like HD exchange coupled with FTIR allow us to study protein interactions and dynamics with incredible precision.
But as with any scientific journey, the more we learn, the more questions arise. Yeah. So what are some of the big unanswered questions in protein science that FTIR might help us address in the future? What are the next frontiers?
Oh, that's a great question. One area that's particularly exciting is the study of protein folding.
Yeah.
It's one of the fundamental mysteries in biology. How does a linear chain of amino acids spontaneously fold into a specific intricate three-dimensional structure?
Yeah, like molecular origami.
Exactly.
And we know that misfolded proteins are implicated in so many diseases, from Alzheimer's to Parkinson's.
Exactly. So if we can understand the folding process in more detail, it could lead to breakthroughs in treating these devastating diseases.
Right.
And FTIR, with its ability to track structural changes in real time, could be a key player in unraveling this mystery.
So we're talking about using FTIR to watch proteins fold in real time, capturing those fleeting intermediate states that have been so elusive in the past.
That's the dream. And I think we're getting closer to realizing it with the advancements in FTIR technology and data analysis techniques.
What about drug development? Could FTIR play a role in designing new and more effective medicines?
Absolutely. One of the biggest challenges in drug development is understanding how drugs interact with their target proteins.
Right.
FTIR can provide crucial insights into these interactions, allowing us to see it at a molecular level how a drug binds to a protein and triggers a specific response.
So it's like having a front row seat to the molecular drama unfolding within our cells.
Precisely, and that knowledge is invaluable for optimizing drug design.
Yeah.
We can use FTIR to screen potential drug candidates, identify the most promising leads, and even guide the design of more targeted and effective therapies.
Okay, I'm convinced FTIR is one of the most powerful tools in the scientific arsenal, and it's clear that FTIR spectroscopy has a bright future ahead. But before we wrap up, I want to bring it back to our listener.
Okay.
What's the key takeaway here? Why should they care about FTIR?
I think the key takeaway is that FTIR is not just some abstract scientific technique. It's a tool that's helping us understand the molecules that make life possible. That the proteins in our bodies are the workhorses of life.
Yeah.
Carrying out a vast array of functions that keep us some alive and thriving. And FTIR is giving us a deeper understanding of how these amazing molecules work.
Right.
Leading to advancements in medicine, drug development, materials, science, and beyond.
It's truly remarkable how this technique, based on the simple principle of how molecules absorb infrared light, has opened up such a vast world of discovery. And it's exciting to think about what the future holds for FTIR and the insights it will bring.
I completely agree. It's an incredibly exciting time to be working in this field, and I can't wait to see what new discoveries lie ahead.
Well, thank you so much for joining me on this deep dive into the world of FTIR spectroscopy. It's been a fascinating journey, and I hope our listeners have enjoyed it as much as I have.
It's been a pleasure sharing my knowledge and insights with you.
And to our listeners, remember that the show notes include links to all the source material we discussed. So be sure to check those out if you want to delve deeper into this fascinating topic. Keep exploring, keep asking questions, and never stop learning. Until next time, goodbye.